Hello friends and welcome. Today I am going to deliver a lecture on population genetics. Let us first try to have a look at the main objectives of today's deliberation. To try to learn about population genetics, to learn about HardyWenberg equation and genotypic frequencies, explaining in detail linkage disequilibrium and factors creating linkage disequilibrium, to give a general idea about the term haplotype and haplotype construction, and finally discussing in detail allelic frequencies and calculation of allele frequencies from genotype frequencies. Now we will start with the brief idea about population genetics. Population genetics is a subfield of genetics that deals with genetic differences within and between populations and is a part of evolutionary biology. In other words, population genetics is the study of genetic variation within populations and involves the examination and modeling of changes in the frequencies of genes and alleles in populations over space and time. Many of the genes found within a population will be polymorphic, that is, they will occur in a number of different forms or alleles. Mathematical models are used to investigate and predict the occurrence of specific alleles or combination of alleles in populations based on developments in the molecular understanding of genetics, Mendel's law of inheritance and modern evolutionary theory. The focus is the population or the species, not the individual. Studies in this branch of biology examine such phenomena as adaptation, speciation, and population structure. Population genetics was a vital ingredient in the emergence of the modern evolutionary synthesis. Its primary founders were Civil Wright, J.B.S. Haldane, and Ronald Fisher, who also laid the foundations for the related discipline of quantitative genetics. Traditionally a highly mathematical discipline, modern population genetics encompasses theoretical, lab and field work. Population genetic models are used both for statistical inference from DNA sequence data and for proof-disproof of concept. What sets population genetics apart today from newer, more phenotypic approach to modeling evolution such as evolutionary game theory and adaptive dynamics is its emphasis on genetic phenomena as dominance, epistasis, and the degree to which genetic recombination breaks up linkage disequilibrium. This makes it appropriate for comparison to population genomics data. Population genetics begin as a reconciliation of Mendelian inheritance and biostatistics models. Natural selection will only cause evolution if there is enough genetic variation in a population. Before the discovery of Mendelian genetics, one common hypothesis was blending inheritance. But with blending inheritance, genetic variance would be rapidly lost, making evolution by natural or sexual selection implausible. The HardyWenberg principle provides the solution to how variation is maintained in a population with Mendelian inheritance. According to this principle, the frequencies of alleles, variations in a gene will remain constant in the absence of selection, mutation, migration, and genetic drift. Now let us try to learn about HardyWenberg equation. The HardyWenberg equation is a mathematical equation that can be used to calculate the genetic variation of a population at equilibrium. In 1900, G. H. Hardy and W. Wenberg independently described a basic principle of population genetics, which is now named the HardyWenberg equation. The equation is an expression of the principle known as HardyWenberg equilibrium, which states that the amount of genetic variation in a population will remain constant from one generation to the next generation in the absence of disturbing factors. The HardyWenberg equation is expressed as p square plus 2pq plus q square is equal to 1, where p is the frequency of the dominant allele A and the q is the frequency of the recessive allele A in the population. In the equation, p square represents the frequency of the homozygous genotype dominant AA, q square represents the frequency of the homozygous genotype recessive AA, and 2pq represents the frequency of the heterozygous genotype dominant A and recessive A. In addition, the sum of the allele frequencies for all the alleles of the locus must be 1. So P plus Q is equal to 1. If the P and Q allele frequencies are known, then the frequencies of the three genotypes can be calculated using the HardyWenberg equation. In population genetics studies, the HardyWenberg equation can be used to measure whether the observed genotype frequencies in a population differ from the frequencies predicted by the equation. Genetic variation in population can be analyzed and quantified by the frequency of alleles. Two fundamental calculations are central to population genetics: allele frequencies and genotype frequencies. Genotype frequency in a population is the number of individuals with a given genotype divided by the total number of individuals in the population. In population genetics, the genotype frequency is the frequency or proportion, that is 0 is less than f and f in turn is less than 1 of genotypes in a population. Although allele and genotype frequencies are related, it is important to clearly distinguish them. Genotype frequency may also be used in the future for genomic profiling to predict someone's having a disease or even a birth defect. It can also be used to determine ethnic diversity. Now let us discuss the next part of our today's lecture, that is linkage disequilibrium. In population genetics, linkage disequilibrium is the non-random association of alleles at different loci. Loci are said to be in linkage disequilibrium when the frequency of association of their different alleles is higher or lower than what would be expected if the loci were independent and associated randomly. Linkage disequilibrium is influenced by many factors, including selection, the rate of recombination, the rate of mutation, genetic drift, the system of mating, population structure, and genetic linkage. As a result, the pattern of linkage disequilibrium in a genome is a powerful signal of the population genetic processes that are structuring it. In spite of its name, linkage disequilibrium may exist between alleles at different loci without any genetic linkage between them and independently of whether or not allele frequencies are in equilibrium, not changing with time. Furthermore, linkage disequilibrium is sometimes referred to as gamete phase disequilibrium. However, the concept also applies to asexual organisms and therefore does not depend on the presence of gametes. Suppose that among the gametes that are formed in a sexually reproducing population, allele A occurs with frequency Pa at one locus, that is Pa is the proportion of gametes with A at that locus, while at the different locus allele B occurs with frequency Pb. Similarly, let Pab be the frequency with which both A and B occurs together in the same gamete, that is Pab is the frequency of the AB haplotype. The association between the alleles A and B can be regarded as completely random, which is known in statistics as independence, when the occurrence of one does not affect the occurrence of the other, in which case the probability that both A and B occur together is given by the product Pa/Pb of the probabilities. There is said to be a linkage disequilibrium between the two alleles whenever Pab differs from Pa/Pb for any reason. The level of linkage disequilibrium between A and B can be quantified by the coefficient of linkage Dab, which is defined as Dab equal to Pab minus Pa/Pb, provided that both Pa and Pb are greater than 0. Linkage disequilibrium corresponds to Dab not equal to 0. In the case Dab equal to 0, we have Pab equal to Pa multiplied by Pb, and the alleles A and B are said to be in linkage equilibrium. The subscript Ab on Dab emphasizes that linkage disequilibrium is a property of the pair A and B of alleles and not of their respective loci. Other pairs of alleles at those same two loci may have different coefficients of linkage disequilibrium. Linkage disequilibrium in asexual populations can be defined in a similar way in terms of population allele frequencies. Furthermore, it is also possible to define linkage disequilibrium among 3 or more alleles. However, these higher order associations are not commonly used in practice. The coefficient of linkage disequilibrium D is not always a convenient measure of linkage disequilibrium because its range of possible values depends on the frequencies of the alleles it refers to. This makes it difficult to compare the level of linkage disequilibrium between different pairs of alleles. Lewontin suggested normalizing D by dividing it by the theoretical maximum differences between the observed and expected allele frequencies as follows: D dash equal to D divided by D minimum. Now let us try to learn about factors creating linkage disequilibrium. There are 5 processes that can produce linkage disequilibrium in a population: epistatic natural selection, mutation, random drift, genetic hitchhiking, and gene flow. Although natural selection is the most important, some of the others, notably gene flow, can create substantial levels of disequilibrium. Number 1 is mutation. Similar to its weak effects on allele frequency change, the process of mutation does not lead to any substantial disequilibrium. Recurrent mutation will certainly produce non-random associations between alleles at different loci. However, recombination typically occurs at higher levels than mutation, and it will break apart any non-random associations of alleles. Furthermore, recurrent mutations are not expected to be associated with the same alleles at other loci. However, in non-recombining regions of the genome, such as part of the human Y chromosome, mutation can form linkage disequilibrium between loci that will not decay and can increase in frequency by drift or selection. Next is random drift. Non-random associations between alleles at different loci may be produced by random drift. Multi-locus models examining the formation and decay of disequilibrium by drift have shown that the expected value of disequilibrium among replicate populations is 0. This is the same result as the expected allele frequency change for alleles at a single locus. However, in any one population, the magnitude of disequilibrium may indeed be substantial. In a finite population with effective population size, an E and a rate of recombination R between loci, the expected value of R square is E multiplied by R square is almost equal to 1 divided by 1 plus 4 NEC. This equation shows that if 4 NEC is small, the expected value of R square approaches 1.0. As 4 NEC increases in magnitude, the expected value of disequilibrium approaches 0. Next is gene flow. Gene flow can produce significant levels of disequilibrium in a population. However, this will occur only when the frequencies of alleles at both loci differ between the populations. The greater the allele frequency differences, the greater the disequilibrium produced by gene flow. If there is linkage disequilibrium between loci in the source populations, then this will contribute even further to the non-random associations of alleles. Population subdivision has the effect of reducing the rate of decay of linkage disequilibrium. If gene flow is small, then the rate of decay is determined by the magnitude of M, the proportion of migrants. If gene flow is more extensive, then the decay is determined by the recombination rate. Now let us try to learn about haplotype construction, 2 loci using SNPs and or microsatellites. A haplotype, haploid genotype, is a group of genes in an organism that are inherited together from a single parent. A haplogroup is a group of similar haplotypes that share a common ancestor with a single nucleotide polymorphism mutation. Mitochondrial DNA passes along a maternal lineage that can date back thousands of years. There are, however, several specific definitions of the term being used in genetics. First, it is a portmanteau word for haploid genotype, which is a collection of specific alleles, that is specific DNA sequences in a cluster of tightly linked genes on a chromosome that are likely to be inherited together. That is, they are likely to be conserved as a sequence that survives the descent of many generations of reproduction. A second specific meaning of the term haplotype, a set of single nucleotide polymorphisms, that is SNPs, on one chromosome that tend to always occur together, that is, they are associated statistically. It is thought that identifying these statistical associations and few alleles of the specific haplotype sequence can facilitate identifying all other such polymorphic sites that are nearby on the chromosome. Such information is critical for investigating the genetics of common diseases, which in fact have been investigated in humans by the international HapMap project. Next is haplotype resolution. An organism's genotype may not define its haplotype uniquely. For example, consider a diploid organism and two biallelic loci, such as SNPs, on the same chromosome. Assume the first locus has alleles A or T and the second locus G or C. Both loci then have three possible genotypes, AA, AT and TT, and GG, GC and CC respectively. For a given individual, there are 9 possible configurations haplotypes at these two loci as shown in the Punnett square. For individuals who are homozygous at one or both loci, the haplotypes are unambiguous, meaning that there is not any differentiation of haplotype T1, T2 versus haplotype T2, T1, while T1 and T2 are labeled to show that they are the same locus, but labeled as such to show it doesn't matter which order you consider them in, the end result is 2 T loci. For individuals heterozygous at both loci, the gametic phase is ambiguous in these cases. You do not know which haplotype you have, for example TA versus AT. The only unequivocal method of resolving phase ambiguity is by sequencing. However, it is possible to estimate the probability of a particular haplotype when phase is ambiguous using a sample of individuals. Given the genotypes for number of individuals, the haplotypes can be inferred by haplotype resolution or haplotype phasing techniques. These methods work by applying the observation that certain haplotypes are common in certain genomic regions. Therefore, given a set of possible haplotype resolutions, these methods choose those that use fewer different haplotypes overall. The specifics of these methods vary. Some are based on combinatorial approach, for example parsimony, whereas others use likelihood functions based on different models and assumptions such as the Hardy-Weinberg principle. The coalescent theory model are perfect phylogeny. The parameters in these models are then estimated using algorithms such as the expectation maximization algorithm (EM), Markov chain Monte Carlo (MCMC), or hidden Markov models (HMM). Microfluidic whole genome haplotyping is a technique for the physical separation of individual chromosomes from a metaphase cell, followed by direct resolution of the haplotype for each allele. Now at last, let us discuss the last part of our today's lecture, that is allelic frequencies. The allele frequency or gene frequency represents the incidence of a gene variant in a population. Alleles are variant forms of a gene that are located at the same position or genetic locus on a chromosome. An allele frequency is calculated by dividing the number of times the allele of interest is observed in population by the total number of copies of all the alleles at that particular genetic locus in the population. Allelic frequencies can be represented as a decimal, a percentage or a fraction. In a population, allele frequencies are a reflection of genetic diversity. Changes in allele frequencies over time can indicate that genetic drift is occurring or that new mutations have been introduced into the population, given the following. Number 1. A particular locus on a chromosome and a given allele at that locus. Number 2. A population of n individuals with ploidy small n, that is, an individual carries n copies of each chromosome in their somatic cells. For example, 2 chromosomes in the cells of diploid species. Number 3. The allele exists in I chromosomes in the population, then the allele frequency is the fraction of all the occurrences I of that allele and the total number of chromosome copies across the population I divided by small n multiplied by capital N. The allele frequency is distinct from the genotype frequency, although they are related, and allele frequencies can be calculated from genotype frequencies. In population genetics, allele frequencies are used to describe the amount of variation at a particular locus or across multiple loci. When considering the ensemble of allele frequencies for a large number of distinct loci, their distribution is called the allele frequency spectrum. That is all about today's lecture. Hope you have understood it well. Thank you very much and goodbye.